Welcome to The Debate. Today we are opening the case file on perhaps the most consequential detective story in human history. We're looking at the origin of SARS-CoV-2. Since late 2019, this virus has reshaped our societies, our economies, our lives, but the question of how it got here has split the scientific community and, well, the public into two deeply entrenched camps. Was this a natural catastrophe, a spillover from the wildlife trade at the Huanan seafood market? Or was it a scientific accident, a spark that jumped from a laboratory bench in Wuhan?
It really is the question that looms over everything, because the answer dictates what we do next. If this was nature, we're fighting a war against ecology and, how we interact with the wild. But if this was science, if this was us, then we have to face the terrifying possibility that the very research meant to protect us actually endangered the entire species.
Exactly. The stakes couldn't be higher. And just to frame this for our listeners, we aren't here for political finger-pointing or internet rumors. We are here to look at the hard evidence found in the source material. I'll be arguing the case that the data, the maps, the genes, the animals, points overwhelmingly to a natural zoonotic event at the Huanan market.
And I will be taking the opposing view. My argument is that the forensic coincidences, the specific biological fingerprints on the virus, and the documented intentions of the Wuhan labs paint a picture of a research-related origin. We have to look at what was missing from the market and what was hidden in the lab.
Okay, so let's start where the outbreak started, the geography. If you drop a pin on a map of the first known cases of COVID-19, you don't see a random scattershot pattern across a city of 11 million people. You see a bullseye.
You're referring to the Warabay study, right? The one on the early case clusters?
I am. Michael Warabay and his colleagues used a technique called kernel density estimation. Think of it, like a weather map for the virus. When they plotted the residential locations of the earliest 155 cases from December 2019, the heat of that map centered precisely on the Huanan seafood wholesale market. It is a tight, undeniable cluster.
But surely, in a city that size, people congregate in many places. It's a busy market.
Well, that's the critical insight, though. This clustering wasn't just about people who work there. The study found that even early patients with no direct link to the market, people who didn't shop there, didn't work there, didn't know anyone who did, lived significantly closer to it than you would expect by random chance. We're talking a median distance of just over 2 kilometers. If the market was just a super spreader event for a virus that leaked from a lab miles away, you'd expect the unlinked cases to be near other transit hubs, like the Hankow railway station or, major shopping malls. They weren't. They were clustered around the market. The virus didn't just amplify there, it bled out from there into the surrounding neighborhoods.
I admit the map is a compelling visual. It draws the eye. But I think it draws the eye away from something equally important. We cannot ignore the context of Wuhan itself. This isn't just any metropolis. It is the global cockpit for coronavirus research. You have the Wuhan Institute of Virology, the WIVI, holding the world's largest collection of bat coronaviruses. But even closer to your bullseye is the Wuhan Center for Disease Control, the WHCDC.
A different institution entirely, though.
Correct, but geographically critical. On December 2nd, 2019, right at the moment your timeline suggests the silent spread is beginning, the WHCDC moved its laboratory. And where did they move it to? A location just 280 meters from the Huanan seafood market. We are talking less than 3 football fields away.
It is a striking coincidence, I'll grant you that.
Is it just a coincidence? We are talking about a laboratory that specialized in collecting and analyzing bat coronaviruses. We know from the source material that they were physically moving their biological inventory freezers, samples, viral isolates, down the street right next to the market, exactly when the pandemic starts. Moving a lab is chaos. It involves transporting sensitive materials through the city. It is the prime time for a containment breach or an accident. In my view, focusing entirely on the market stalls while ignoring the virology lab next door is a classic case of tunnel vision.
I understand the temptation to link them. It feels intuitive. But proximity is not causality. If the WHDC was the source, we would expect the earliest clusters to be around their researchers' homes or their dormitories or the hospitals they visited. We don't see that. We see the market. And we don't just see cases near the market, we see the virus physically in the market. You mean the environmental swabs taken after the closure?
Precisely. In January 2020, the Chinese CDC went in and swabbed the walls, the drains, the freezers, the cages. They found SARS-CoV-2 RNA heavily concentrated in the West Zone. And thanks to recent metatranscriptomic analysis, basically sequencing every scrap of genetic material on those swabs, like digging through the genetic trash, we now have what I call the smoking gun. In stall 29 specifically.
Stall 29 is the wildlife stall, correct?
Yes. In that specific stall, researchers found a statistically significant mix. They found SARS-CoV-2 RNA. But mixed in the exact same sample, on the same surfaces, they found the DNA of raccoon dogs, masked palm civets, and bamboo rats. These are the exact species we know are capable of carrying and transmitting this virus. We have the vector and the virus in the same square meter of space at the start of the pandemic. Hold on, though. Let's look at what that evidence actually tells us. It tells us a raccoon dog was there and the virus was there. It does not prove the raccoon dog was infected. It is equally plausible, and I would argue likely, that a human, perhaps a patient zero who worked at the nearby lab or lived nearby, went to the market and shed the virus onto those cages. You are seeing the scene of the crime, but you are mistaking the victim for the perpetrator.
But you have to pile assumption on top of assumption for that to work. You have to assume a lab worker got infected, didn't infect anyone at the lab, didn't infect anyone at home, went to the market and specifically contaminated the one stall that held the exact animals capable of carrying the virus. That's a massive bank shot. The zoonosis theory is a straight line. The animals were there, they carried the virus, they affected the handlers. We saw this exact mechanism with SARS-CoV-1 in 2003.
We did see it in 2003. And that contrast is actually the strongest point for my side. In 2003, within months, we found the infected civets in the markets. We found antibodies in the animal traders. We had the chain of custody. Here, Chinese authorities tested 80,000 animals from around the country. They tested the market animals. They found nothing. Zero.
That is a fair critique, but we have to remember the timeline. The market was shut down and sanitized on January 1st. The animals were largely disposed of before they could be properly sampled.
But 4 years later, we still haven't found the upstream farm. If this was a natural trade of infected animals, where are they? It feels like the zoonosis argument is relying on a ghost animal that vanished into thin air. Meanwhile, the ghost in the machine, the virus itself, has features that look incredibly suspicious.
You're referring to the biological structure of the virus.
I am. I want to talk about the blueprint. In 2018, the EcoHealth Alliance and partners at the WIV submitted a grant proposal to DARPA called Diffuse. It is chilling to read today. They explicitly proposed going into bat caves, finding SARS-related viruses, and inserting a specific genetic feature called a furin cleavage site, or FCS, to see if it would make them more infectious to humans.
And SARS-CoV-2 has a furin cleavage site.
It is the only virus in its immediate family of SARS coronaviruses that does. It is the feature that made COVID-19 so transmissible. So we have a proposal in 2018 to put this exact engine into a bat virus in Wuhan. 2 years later, a bat virus with this exact engine appears in Wuhan. If I write a plan to rob a bank, and 2 years later the bank is robbed exactly according to my plan, am I just unlucky, or am I a suspect?
It's a powerful narrative, I agree. But biology is messier than a bank heist. The presence of a furin cleavage site isn't impossible in nature. Other coronaviruses, like HKU1 or MERS, have them. They evolve through recombination. Imagine that market again. Cages stacked on top of each other, animals urinating and defecating on one another. It is a viral blender. Recombination happens there.
But look at the code, not just the feature. The genetic code for the FCS in SARS-CoV-2 uses a specific sequence, CGG-CGG. This codes for the amino acid arginine. Now, in coronaviruses, that specific double CGG combination is extremely rare. But in human laboratories, it is the preferred code. It is what you use when you are doing codon optimization to make a virus work better in human cells. The diffuse proposal actually mentions codon optimization. The virus literally carries the signature of laboratory tools.
I'm wary of that signature argument because it relies on what we call a no-seum approach, claiming that scientists created a virus that looks natural but isn't. But to engineer a virus, you need a backbone, you need a template. The closest known virus of the WIV was RATG13. It is 96.2% identical to SARS-CoV-2.
That sounds very close.
In evolutionary terms, it's actually miles away. That 4% difference represents decades of evolutionary divergence. You cannot turn RATG13 into SARS-CoV-2 without leaving massive genetic scars, which simply aren't there, unless you are suggesting they had a backbone they never published.
That is exactly what I am suggesting. And this brings us to the data blackout. We know that on September 12, 2019, the WIV took their main viral database offline. It contained 22,000 samples. We know they were working with unpublished viruses from the Mojiang mine, where miners had died of a mysterious SARS-like pneumonia. The dark matter of their collection is exactly where the progenitor backbone would be hiding.
Institutions hide data for many reasons: bureaucratic fear, embarrassment, national security that don't equate to guilt. But I want to counter your missing backbone with a piece of evidence that I think is even harder to explain for the lab leak theory. I want to talk about lineage A and lineage B.
The two early branches of the virus.
Correct. When we sequence the very earliest cases, we see two distinct families of the virus circulating simultaneously. Lineage B was the one linked most heavily to the market. Lineage A appeared slightly later in the data but is evolutionarily older. Now, Pecker and colleagues ran phylogenetic simulations, basically rerunning the pandemic's start thousands of times on a computer.
Okay, and what did the simulation show?
It showed that if this were a lab leak, a single person getting infected and walking out the door, the chances of generating two distinct lineages like A and B immediately are vanishingly small. A single spark usually creates a single fire. But if you have a scenario where multiple infected animals are being sold in a market over a period of weeks, the model fits perfectly. It suggests at least two separate spillover events. One animal infects a human with lineage B. A few days or weeks later, another animal infects someone else with lineage A.
The double spillover theory.
Yes, it sounds improbable until you realize that's exactly how nature works. If a farm is infected, they are shipping cages of infected animals repeatedly. It's not a single spark, it's a bombardment.
I have to push back on that. You are asking us to believe that after decades of no SARS spillovers, nature rolled the dice and hit the jackpot twice in the same city, in the same distinct time frame. That strikes me as statistical, desperate pleading to save the hypothesis. Isn't it more likely that the virus was evolving inside a human population, perhaps circulating silently among lab workers before it hit the market?
If it was circulating silently, we would have seen it in the hospitals. The retrospective search of 76,000 medical records in Wuhan found nothing prior to December. No hidden wave of pneumonia. The signal starts loud and clear at the market.
But did it? Stoyan and Chu published a critique recently that attacks that very certainty. They argue ascertainment bias. In December, doctors were specifically told, if you see a patient with pneumonia, ask if they've been to the market. It's the classic problem of looking for your keys under the streetlight because that's where the light is. If you only test market-linked people, you only find a market cluster.
That bias existed for a short window, yes, but remember the Wura-based study I started with. It specifically filtered for that. It isolated the cases that had no history of the market. These were people who didn't know the market was the danger, and yet their locations still mapped to that neighborhood. You can't explain that away with bias. That is the virus moving through the community, radiating out from the epicenter.
Let's pivot back to the institution itself. We've talked about the geography and the genetics, but we haven't talked about the safety culture. There is a misconception that this work was done in spacesuit labs, BSL-4.
The WIV has a BSL-4 lab, yes.
It does. But the records and the source material confirms this, show that the work on bat coronaviruses, including the creation of chimeric viruses, was largely done in BSL-2 laboratories.
For our listeners, explain the difference.
BSL-4 is the spacesuits with independent oxygen. BSL-2 is a standard dentist's office. You wear a lab coat, gloves, maybe simple eye protection. Crucially, BSL-2 usually relies on a biosafety cabinet, but the room itself isn't necessarily under negative pressure to contain airborne viruses. We know they were testing viruses like WIV1 and rat TG13 at this level because it was faster, cheaper, and easier.
And that is standard practice in many parts of the world, though. If a bat virus hasn't been shown to infect humans yet, it is often handled at lower containment to facilitate the speed of research.
And that is the hubris that leads to disaster. The entire point of the research was to see if these viruses could jump to humans. If you are testing for human infectivity, you should treat the sample as if it is infectious. The intelligence community assessment noted that researchers at the WIV fell ill in autumn 2019 with symptoms consistent with COVID-19.
Consistent with is a very broad brush, though. It's also consistent with the flu, which is rampant in Wuhan in November.
Perhaps. But when you combine sick researchers, a database deleted in the middle of the night, a request to retract sequences, and a chaotic laboratory move 280 meters from the outbreak site, the constellation of events is damning. You argue from affirmative evidence, the raccoon dog DNA. I argue from the evidence of intent and the evidence of cover-up. The diffuse proposal provides the intent. The behavior of the WIV suggests the cover-up.
I see it differently. I think the lab leak theory relies heavily on the absence of evidence, the missing backbone, the missing patient zero. The zoonosis theory relies on what is actually there. We have the market, we have the animals, we have the lineages. Nature is the most powerful bioterrorist we know. It has done this before with SARS, with MERS, with Ebola. To think that this specific time, in a market filled with known vectors, it was actually a wizard behind the curtain, it violates Occam's razor.
I would argue Occam's razor cuts both ways. What is simpler? That nature performed a complex double spillover event with a virus perfectly primed for human transmission, leaving no trace in the animal population, or that the people specifically asking for funding to create that exact virus, working in the same city, made a mistake.
It seems we are at a fascinating impasse. We are both looking at a puzzle with missing pieces.
We are, and I think that is the honest conclusion here. The polarization of this topic has forced people to choose a team, but the data is actually quite complex.
So let's try to summarize where we stand for our listeners. What is the core case for the lab-associated origin?
I think it comes down to means, motive, and opportunity. The means, BSL-2 labs handling airborne pathogens and using reverse genetics technology. The motive, the diffuse grant explicitly seeking to put furin cleavage sites into bat viruses. And the opportunity, the WHL-CDC moving its lab right next to the market, and the unexplained illnesses in the autumn. Without a natural host scout after 4 years of searching, the most parsimonious explanation is that the scientists looking for the pandemic accidentally caused it.
And the core case for zoonosis?
Geography, genomics, and ecology.
Exactly. The geography. The cases clustered tightly around the market, not the labs. The genomics, the two lineages, A and B, which strongly implied two separate natural spillover events, a phenomenon incompatible with a single lab leak. And the ecology, the presence of raccoon dogs and civets in stall 29 mixed with the virus, mirroring the exact mechanism of SARS-CoV-1.
So what would it take to settle this, to end the debate?
To prove zoonosis definitively, we need that sample from an upstream form. We need to find the virus in an animal in 2019, showing the progression to humans.
And to prove the lab leak definitively, we need a brave whistleblower. Or we need that deleted database to be recovered, showing the progenitor sequence, sitting in the WIV's hard drives prior to December.
Until then, we must follow the evidence wherever it leads, with humility. This isn't just about being right, it's about stopping the next one.
Absolutely. Whether it's better market regulation or stricter lab safety, we have work to do.
Thank you for joining us on The Debate.
Keep questioning.